Good afternoon, everyone. You can hear me, Parissa? Great. So happy to see so many of you from so many different units across campus. It's wonderful to have you all here today for this BRACE seminar. So excited to have with us Dr. Jen Provencher. So she's going to be speaking to us about plastic pollution as an emerging environmental contaminant. And let me tell you a little bit about Dr. Provencher. She's a research scientist with the Ecotoxicology and Wildlife Health Division in Environment and Climate Change Canada. She has a BS and a B of Education from the University of British Columbia, her master's degree from the University of Victoria, and her PhD from Carleton University. So her postdoctoral work was done at Acadia University, where she was a Weston Fellow and a Liber Aero Fellow. Dr. Provencher has collaborated with partners across the Arctic since the 2007-2008 International Polar Year, when she was a graduate student and visited the Arctic for the first time. Her current work addresses wildlife health, and in the Arctic it currently focuses on legacy and emerging contaminants, including plastic pollution that we're going to hear about today. I also wanted to mention that given her wildlife health mandate, really interesting that more recently her research program also includes working with partners on emerging wildlife health issues such as SARS-CoV-2 in wildlife. She is currently the lead of the long-term seabird contaminants project under the Northern Contaminants Program, and is the co-chair of the Litter and Microplastics Expert Group for Canada under the Arctic Council's Arctic Monitoring and Assessment Program. So please join me in welcoming Dr. Provencher, and thank you so much, Jen.
Awesome, thank you. Hi everyone, super excited to be here. I'm looking forward to this talk. Just a few things before I start. I tend to wander, but I'm going to try really hard. But if I'm walking around, you got to hit me back to the front because they're recording it and I have to stay attached to this leash here. If at any time you can't hear me because I'm too quiet, not often, too loud, sometimes, you got to flag me down. Because if you don't understand what I'm saying, I'm not doing my job properly. All right, so you gotta keep me in check because I can just run off and ramble and get excited. So if something doesn't make sense, flag me down. So I'm going to start with a little bit about wildlife health. So my job is wildlife health, and so this is kind of a tricky thing to define sometimes. My group generally works on a range of topics. So we do talk about plastic pollution in seabirds a lot, but not always. We do actually do a lot of disease work, so this is SARS-CoV-2. Currently in North America, we are in the largest outbreak of avian influenza ever recorded in North America. So a lot of my team right now is focused on avian influenza dynamics and looking at that. My lab also focuses on oiling in birds. Not to this degree in terms of oiled birds who then show up dead, but what happens when birds and wildlife are exposed to these low-level chronic layers or levels of oil pollution. So we do a lot of work on that. Additionally, people in my group and some of my grad students who I support work on things like entanglement in plastics. We do some work on fisheries bycatch, so what happens when birds are caught incidentally in fisheries, and then of course harvest. So all of these things kind of add up in what we work on, but I'm only going to talk about plastic pollution today. We're not going to talk about any of the other things. So to kind of put it in context, this is what I work on. And I just recently did this based on some of the papers that we work on. And the answer is I work mostly on seabirds, mostly on plastic pollution and additives, mostly in the Arctic.
Excuse me?
Yes?
Yes, sorry. You're not sharing your screen, so...
Oh.
I'm so sorry. I really didn't mean to interrupt you guys, but...
No, it's fine. It'll be in the recording after.
It will be in the recording after I am being told. It's not going to happen in real time.
Sorry, we're running the Zoom and the presentation off of different computers, so it's connected to a different integrated camera, so I can't share the screen with the presentation. It's on a different computer basically. So we'll share the presentation afterward, the slides themselves.
Okay, thank you.
Okay. So this is what we're going to talk about today. But again, just as I was talking to some of the grad students, being a government scientist, we often kind of get yanked off of other things when there's an emerging concern, right? So we haven't done a lot of plastic pollution work this year, but we sure have done a lot of avian influenza work. And so there is no hard and fast rule on what I work on, but this is kind of the area I get to play in generally. Okay, so wildlife health, just a little kind of, because most people think about conservation, and this is where most of our like wildlife work is done, right? But wildlife health kind of comes in when you have an intact habitat, right? Your forest is healthy, it's present, your kelp forest is present, your meadow is present. This is where a lot of my conservation ecology colleagues work in, right? And then of course there's a sustainable harvest question, but if your habitat's intact and your harvest is sustainable, but you're still having a problem, that's when you land on my desk. And that's why you have a bit of a catch-all. Parasites, pathogens, contaminants, infectious disease, zoonosis, all these things, that's what my group works on. So we kind of cover a wide range of issues. But today we're going to talk about plastic pollution. And then in the world of plastic pollution, size matters, right? There's a lot of different plastic pollution. And some people work exclusively on these kind of big macroplastics. Sometimes they even go up to like megaplastics. This can include things like even discarded fishing nets, which can reach as a single kind of piece of macro litter, right? Tons, literally a ton of debris, right? But a lot of our work sits here as this kind of micro mesoplastics, and then we do have colleagues that kind of go down into this ultra fine level, and these are not clear. This is purposely not clearly bracketed plastics because this is a discussion topic of what do we call these different size classes. And there are a lot of people kind of actively thinking about this. So just to kind of give you a primer on plastics pollution. So I first started working on plastic pollution when I was working on my master's. I was given a bunch of harvested seabirds from the Arctic and told to quantify their diet. And this was in the International Polar Year for 2007 and 2008. And what they wanted me to do was compare the diet in the birds to the 2007, 2008 back to the historic samples that we also had, which were from the 1970s and the 1980s. And through that, I started to find plastic pieces. And so my thesis supervisor, though I'm not sure he would, he loved it in the moment, allowed me to shift part of my thesis to actually looking at microplastics. And so I kind of had to think about what that looked like and what those definitions were. So there's two types of, we talk about marine debris a lot, but it's really plastic pollution generally. And so plastic pollution can kind of come in these pellet forms. These are industrial pellets or nurdles, right? These are the pre-products of all the other things, right? So oil and natural gas, right? Polymers come out of, natural gas and oil come out of the ground. It gets partitioned off. There's a piece of that that's partitioned off, forms into these pellets, right? There's a lot of additives and colors and all kinds of things added. These are the industrial pellets, right? They get shipped all over the world to plastic factories who then make all things consumer, right? So this would be the chairs we're sitting on, the tables we're sitting at, all the litter you walk by, and the garbage, right? That's all user plastics. The EPA, the Environmental Protection Agency in the United States, has estimated that about 20 billion pounds of plastics enter the ocean every year. And so someone once did the calculation that is essentially a dump truck of plastics kind of entering the ocean every minute. So I've been talking kind of for 7-8 minutes by now. You can imagine 7 dump trucks like backing up and just like offloading into the ocean. That's the equivalent. So it's kind of this constant flow. Whether or not that figure is still current, because that's about 10 years old, is debatable. Some say that's gone up, some say that's gone down. It probably depends on your jurisdiction and what that actually looks like. So this is a crazy slide, and the purpose is not to kind of for you to read it all, but in response to kind of the plastics problem, the government of Canada a couple years ago came up with the Canadian Science Plastic Agenda. And so I'm a government scientist, so I'm going to kind of link back on like what that means to be a government scientist and working in this space. So as a government scientist, I get told what to work on. I don't necessarily get told how to work on it, right? But I get told a theme, right? I work for Environment Canada. I can work on migratory birds. I can't work on fish, right? That's DFO, right? So I get a theme. The theme that I'm kind of tasked with is plastic pollution. And so part of kind of the government of Canada's goal to kind of figure out internally what we can do, and then how do we do this externally with partners, is to develop this kind of plastics agenda. And so this is available online. There's lots of pretty graphics, but there's 2 pillars of 5 that basically drive all of my work on plastics. And one of them is this kind of method program, right? Is the detection, quantification, and characteristics of plastics in the environment. And then there's this impact on wildlife health. And so this always drives and always comes back to all of the questions that I kind of have free reign to investigate and explore. So in the plastics world, in my lab, we have 3 main kind of themes. And I'm going to start with the ingestion and accumulation. I'm going to move through really quickly, though I could talk about this for a whole hour. This idea is animals as vectors or transformers of plastics, but then I'm going to land on this idea of plastics as a vector for contaminants. So seabirds are kind of the poster child for plastic pollution, right? Put up your hand if you've ever seen a picture of a seabird's stomach kind of exploding with Bic lighters. Only 2 hands? Yeah, there's more than that, right? This is like the common thing. But one of the things that's really funny is that seabirds have always been kind of vulnerable to ingesting anthropogenic debris. And so when we go back to the records, there's actually this report in 1838 of a Wilson storm petrel, which is this bird right here, and they're quite tiny. And it's this little bird and that have ride these currents, and they do this little tiptoeing thing across the surface of the water, and they're feeding on the zooplankton and the fish that are at the top. But unfortunately, they also eat other things. And so there's a report of a candlestick inside the stomach dating back to the 1800s. So they're vulnerable just on how they eat to this kind of stuff that floats at the surface. So even dating back, and so we talk about plastics as an emerging contaminant. This is not a, this is a debatable topic, but it's not really emerging. Right? It actually is recognized in legislation, specifically in the London Dumping Convention and the Marpol Convention back to the 1970s. They actually put legislation in around dumping at sea and plastic pollution, recognizing it was always a problem. Specifically in the North Sea, the Oslo-Paris Conventions, or the OSPAR, actually identified the northern fulmar, which is a seabird specifically, as the environmental indicator for plastic pollution in the environment. And so a lot of this early work on birds and plastic pollution really is in this North Sea area. And I usually don't put up like big quotes like this, but this is really specific to the legislation, and it talks about the species, the kind of minimum or the kind of more than what they should have less. It talks about 10% of them should have this amount or less. They talk about the sample size, they talk about the number of sites, and then they talk a time period. And so when they're building it into the legislation, they have all of these things defined that it has to be done in this particular way. So it's not just about, well, you should study northern fulmars. You should be studying fulmars from at least this many sites over this many time period, and this is what the quality indicator is. And so this is what we call as the ecological quality objective. And so the goal is for 10% of the birds to have 0.1 grams or less in those birds, and that's the ecological goal that they're trying to get to. And they actually can measure that over time. And Jan van Vronniger's lab out of Texel in the Netherlands is the leader of this project. They've been working on this since the 1990s, and we've worked with them to look at how declining trends in the North Sea and in relation to Canadian levels actually are projected to decrease over time given the kind of current trajectory. And it looks like, again, according to this kind of modeling, is that it's probably going to take till about the year of 2050 until we get to that ecological quality objective. And so this is kind of what the European Union is thinking about in terms of like what the current levels are, what are the policy changes that we need to get to, and then where do we think we're going to cross that finish line? And do we need to speed it up, right? Because it's using this to say, well, is this kind of current rate acceptable or not? And so we have fulmars in Canada. We have fulmars in all 3 coasts. They're kind of an offshore species. Anyone actually seen a fulmar before? Oh, like 3 people? So if you don't go out at sea, you often are not going to see them unless you go to places like Iceland and the UK and Ireland, where weirdly you just like drive up to their colonies and they're just kind of flying around. In Canada, you have to take like multiple planes, multiple boats to get there. But we do have fulmars on all 3 of Canada coastlines. So part of the work that we've been doing for the last 15 years is actually doing plastic pollution assessment in exactly the same way as our European colleagues. So we can actually monitor and have harmonized data together and standardized data to actually look across those sites. Okay, so this is where you all have to do some work. So we have 4 sites that we've monitored in time for plastic pollution. Prince Leopold Island, which is way up here in Lancaster Sound, Vancouver Island on the west coast, the Labrador Sea in between Newfoundland, or Labrador and Greenland, and then Sable Island, which is a tiny island. Anyone know about the Sable Island horses? You probably know about them. There's also fulmars on that island. Well, at least they wash up, and so there's the Sable Island site there. So of these 4 samples, sites, what I want you to do is think about what site do you think will have the highest level of plastic pollution in the fulmar stomachs compared to each other. So I'm going to give you a minute. I want you to think about it, and then you're going to actually discuss it with the person beside you if you would like. What site has the highest level of plastic pollution in fulmars, and what site has the lowest levels of plastic pollution in fulmars? Using what you kind of know about where urban areas are, and ocean currents, and air currents, I want you to tell me what you think. I'm going to give you all a minute to think about it. What site has the highest? Which site has the lowest? Nope, hold. I want everyone to think about it. Juliet, you can't answer. One of my grad students is in the back, and she's banned from answering any of these questions. Okay, I want you to talk about it with the person beside you for a second. And I want you to explain what is the highest, what's the lowest, and why you think that. Do you have a question? Okay. Okay, I'm going to give you 5 seconds to wrap up. All right, who's willing to share an answer with the group on what you think is the highest and what you think is the lowest? No one? Yes.
The highest is Prince Leopold Islands.
Why?
I guess there is accumulation, there are less currents, and so there's no way of getting rid of it when it is there.
Okay.
And so by that reasoning, the lowest would be Vancouver Island.
Okay. Any other answers?
We were thinking Sable Island might have the least, just because of the size and frequency there of birds.
Okay, so that's about the birds.
Oh, there.
Anyone else? Anyone have any different answers?
The maximum might be at the Vancouver Island because it seems close to the GPGP, the garbage patch of plastic.
The North Pacific Garbage Patch?
Seems like that.
Okay, and where's the lowest?
Not sure.
Any other answers?
Someone in the chat says the Atlantic, but I'm not sure for what.
Okay, chat person, you gotta tell us, the Atlantic highest or lowest? Let's give him a second.
East Coast, higher.
Higher, okay. Any other questions? Okay, I've heard no right answers. If it makes you feel better, I have given this presentation to ministers of the environment, and they also have not gotten it right. So you are good company. So this is the answer. So Sable Island has the most weight-wise. They have the heaviest plastics, right? Labrador has intermediate. Prince Leopold actually has the lowest from a weight perspective. Interestingly, right, going back to the North Pacific garbage patch, people often think that Vancouver Island has the highest. They're lighter than the Sable Island birds. Like, they have the most kind of weight-wise, but the Vancouver Island birds have the most numerous plastics. So they have more smaller pieces.
So
From numbers, they have the highest, but biologically, weight is the measure, right? The North Sea ecological objective is 0.1 grams or less, right? Because biologically, if you're a bird, what are you evolutionarily designed to do to find your food? Fly, right? Weight matters to birds almost more than anything else. And so the ecological quality objective is weight. So Sable Island ranks number 1, Vancouver Island 2, 3, 4. And so why is this? What we think is so the North, it's a hard map to see, but the North Pacific gyre patch is kind of out here, right? And so what happens is the California current comes in here, California current goes down, and this goes up. So this is actually very far away from the North Pacific garbage patch. But there's also a garbage patch in the Atlantic, right? The North Atlantic garbage patch is actually much closer continentally here. And if you think about this island, what is this island downstream from? Any of the major cities, what is it downstream from? New York, Boston, Pittsburgh. Can't even name them all. They're all on the East Coast, right? This is a massive flow. And the Gulf Stream takes all of that garbage and brings it past here. And the Gulf Stream takes it over here. So much so that one of the most identifiable plastic items in Scotland is actually milk crates from Nova Scotia, right? So we know it travels a long way. Okay, so because we have harmonized our methods and we've standardized our metrics with our European colleagues and our American colleagues, we actually can do this gradient. So in the western North Atlantic, Devon Island is the most northern of these sites, and as we move south, we see this increase at Sable Island. Same thing here in Norway, as they move from Svalbard south into the English Channel, we see these high levels. Same thing here, as we go from Alaska down into California, we see an increase. So it's very clear we have this north-south gradient in kind of 2 ocean basins along this. And so you have the ecological quality objective here, right? They want 10% or lower to have this 0.1 grams, right? And you can see that all of the sites so far are above this ecological quality objective, which is the policy target, except our 2 sites in the Canadian Arctic. So when I look at our data, the first time I ever looked at our data, I was a grad student, I was a master's student, and I was like, oh, we found plastics in birds in the Arctic? Like, oh, this is so devastating. I showed this to my European colleagues, and they were like, that's fantastic. You're below the ecological quality objective, right? And so we saw this as devastating, and they saw this as like inspiring, because it means it exists somewhere. And so this gives them inspiration that even in the Arctic, it can exist. If we were above their ecological quality objective in the Arctic, I suspect they would have changed their ecological quality objective, because they would never have been able to get there. It would be a policy target that would be unrealistic. But they see our data as making their policy target as aspirational and possible. Okay. So in 2020, we actually added the Fulmars to our SESI program, which is very kind of internal, but we do this a lot. It's called the Environmental Sustainability Indicators. Whoops. And so there's a whole set of these indicators that the Canadian government uses to understand if the policies that they're putting in place are actually doing anything, right? Policy is great. Policy without indicator is kind of useless, because you don't know if it's doing anything, right? Policy with no target is kind of just like an academic poli-sci discussion, right? And so part of my job, again, as a government scientist, is informing these environmental sustainability indicators. And so the only plastics indicator on the books currently is the northern fulmar. And so part of my program is making sure that we continue these studies, we do the collections in a harmonized way, we standardize the data, and we kind of keep this bar chart going, right? And so this is a large part of my program, is this long-term monitoring. Okay, but a seabird is not a seabird, is not a seabird, right? And so just because you can find plastic pollution in fulmars, which is here, right? So this is an example of Prince Leopold, that colony up in the high Arctic, and 84-ish% of them had some level of plastic. But these other species had very low levels, 10% for kittiwakes. These guys don't have any plastics in it, as far as we can tell, above 1 millimeter. I always have to put that size classification in there, right? And we think this has to do with their foraging strategy. These guys are divers, right? These guys are feeding at the surface. And then there's the gulls. And we all know what gulls eat. They eat anything that's on the ground, right? And so we have actually looked at gulls, and these are specifically gulls from the dump in St. John's. And they actually have to kill the gulls every year because they are in the flight path of the airport. And so there's a big cull in Newfoundland every year for these gulls. And we actually started to look at their stomachs just to see what the range is. And you can see there's chipboard, there's drywall in here, there's aluminum, there's metal, there's a coffee cup wrapper in there, right? There's all kinds of things. And this is some work that Sahar Safe has done. And we've been looking at this for a couple years now by looking at these 3 gull species. But this actually kicked into a whole different program in light of COVID. What did we see appear everywhere in the litter and in the gutters as COVID developed? Some of them, right, we're still wearing them, right? Masks, gloves, all the PPE garbage. And so because we had these pre-COVID pre-COVID date collections, it was ongoing. So we have these early COVID gulls, and then of course we've got these kind of deep COVID gulls and ongoing. We've actually been able to look at, when we see the type of debris change, and we know this through environmental data, there's a well-documented change in this. Do we actually see it change for the birds? And Carrie Shooten is a PhD student at Guelph University that I co-supervise, has looked at this. And what's really interesting is that we don't see any ingestion. So just because it's in the environment doesn't mean they're actually ingesting it, even in the dump, which is kind of crazy to me. But what we have found is it's actually started to show up in their nests. And so this is a little hard to see, but this is a cormorant nest. There's 2 chicks. There's one chick kind of facing this way. There's another little head here. There's a couple of eggs. And then we've got a PPE mask in there. And so she's done a lot of work at quantifying how PPE garbage is kind of being collected and concentrated in the environment related to PPE. So again, I'm going to put you to work. I can't do all the talking, all the work up here. So she's been going out and looking at nests across Lake Ontario, and she's actually been quantifying where she finds PPE garbage and where she doesn't find PPE garbage. So I would put you all to the test, and I want you to look for the PPE garbage in these sites or these nests. And how many nests do you see that have PPE garbage in them? So when you're ready, I want you to show me on your hand. Is it 1? Is it 5? Just put up your hand. You don't have to put a height, but just like somewhere, show me your hand by the numbers that you have. And I am judging you all, whether you answer or not. So just for scale, so you know, right? This is a foot, right? This is someone's boots. Okay, once I start seeing hands with answers, I'll go on. But I don't see any hands with any answers right now. Okay, you ready for the answer? Oh, still discussing. 4. Hard? Difficult? Easy? What do you guys think? Very difficult. Very difficult. So part of Carrie's work has actually done these photographs A portion of them, so some of them she photographed, some of them, and then she did like an observed in the moment, like being able to look around it. And then some of them she took back to the lab, deconstructed, and actually looked at. And she found that if 100% is what she kind of captures in the nest, if you're able to kind of be there and look around and do observations, you can get 50% of the items. And if you only have a picture to stare at, which is what I just made you guys do, you only get about 10%. And so this is one of the things that we're kind of thinking about on leading as we, again, Environment Canada, right? As we track this new pollution in the environment, the fate and effects of it, this is kind of the things that we're not struggling with, but we're having to work through, right? How do we actually track and monitor this? Okay, animals as vectors of plastics. This is a piece that I love. I'm not going to spend a lot of time on it today. We have done some seabird work on it. But some of the stuff that is really exciting, and I put this in for like the nano peeps in the room, is some of this work that we're doing with crickets. And this is actually with Heath McMillan's lab at Carleton University, who I co-supervise a bunch of students with. And so Heath, not a plastics person, but he's got insect models, and he does like cold tolerance, thermal tolerance type work. And so he has these colonies of crickets, and I was like, well, we should just like add microplastics to one of their diets and see what happens. And so what we did, and Marshall Ritchie is the master student who's been working on this, is we've been feeding microplastics, specifically 100 micron blue fluorescent polyethylene beads. And so you can see this is the cricket. You can actually see it's glowing there. So we specifically picked fluorescing beads so that we could track them, because this is a fate study. We want to figure out where the plastics go effectively. And so it's a little tough to see, but this is actually a gastrointestinal tract of a cricket. Don't ask me how they dissect out a gastrointestinal tract of a cricket. It's very carefully done. I'm used to working on birds you can like hold as a football, and these are like the tiniest little things. But you can see, so here's an example of a bead. This is an intact bead, but then you have all of these little pieces. And so what we're finding, what Marshall's finding, is that as the beads move through the gastrointestinal tract, because the crickets are often eating seeds and grasses, things that need mechanical grinding, they are breaking down the plastics. And so this is what Marshall found. He looked across all the different gut regions, and what he finds is that increasingly you get these like really tiny microplastics. And so his detection limit with the current methodology is about 1.35 microns, which is on our stuff is about 5 pixels. But what he's finding is that there are certain kind of insects in the world that are great transformers of the plastic pollution. And so that's kind of filled in some of our fate questions, but then what that really kicks into, right, is what the effects are. It's like, so what, right? But that's like a whole different spectrum. But we have a study where we're actually looking at how seabirds are physically moving plastics around the environment, how marine mammals are moving plastics around the environment, and of course on these micro scales, how insects are actually moving the microplastics around agricultural fields. But I'm not going to talk about that. I'm going to talk more about the contaminants, because I know some of that is of interest. So plastics as a vector for contaminants is something that we have worked on, and we've kicked it up in gear over the last couple of years, and I'll tell you why. So first of all, plastic ingestion. So to kind of go back to seabirds, we have done this work where we've actually looked at the polymers in the birds, right? So it's like, okay, they're ingesting plastics. Does it matter? What does that actually mean? And so what we've actually done is, and these are, for some people in this room, these are like really big plastics, right? Like I can hold it in my hand, you can pick it up with a forceps, it's easy to identify, right? So you can take it to FTIR, you can put the plastics on, and you get this, really pretty spectra, and then you can identify what the different polymers are. And even in this very small group of phalaropes, and these guys have a lot of plastics. They have 100% of the phalaropes that we look at have some level of plastics. They're very, very full of plastics. But where we've done all the plastic, it really emphasizes for us that there are a lot of different polymers, right? And so when we talk about plastics, when I started working on plastics, someone said to me, they're like, yeah, but like, I don't know, my 2-year-old probably has plastics in their stomach right now. Like, does it really matter? Okay, sure, but one of the things, we probably have all eaten food off of plastics that was stored in plastics, right? is it a big deal? But what we're finding with some of our FTIR data or polymer data is that, we, hopefully most people, are eating food, related to food grade plastics, right? There's certain plastics that are approved to kind of whole food, and that's what we're exposed to. But what we find in the birds is they have all kinds of plastic debris and many polymers that have additives, industrial plastics that have different chemicals that are not food approved, right? They're not kind of supposed to be ingested. And so this really opens the door for all of these other things to come in. So it's not just the food grade plastics, it's all this cocktail of both polymers and chemical contaminants that we're concerned with. And so we've done a bunch of work where we've looked at, because my long-term program is looking at legacy contaminants. And so we have things like PCB, DDT, these are legacy contaminants, right? These are like the old Dirty Dozen, Stockholm Convention stuff, right? We've actually looked at whether plastics can be a vector for that type of contaminant. And the answer is yes. So, we've done water samples in relation to plastic particles, in relation to fulmars. And definitely there are, more PCBs on the plastics than their environment, a bioconcentrator, right? But when we look at the the congeners in the plastics in the livers, it doesn't really seem to equal. And the thought behind it is that there's enough PCBs in the environment that they're still getting them through the fish and the water, that a little bit more on the plastics, probably not a big deal, right? Okay, so then we start looking at chemicals of emerging concern. And this is where we start getting into these plastic-specific types of contaminants. And so this is actually work that we did with Xiaolu at UCAR a couple years ago, and we actually looked in eggs of animals and then livers. Apologies. I do not need to pick up my daughter from school today, so I do not need that alarm. This is why, because I can just keep talking and then I miss the school pickup and I get in trouble by the 7-year-old, so I have to set myself an alarm. So we looked at the eggs and we looked in the livers and we looked in kittiwakes, and if you remember way back to a few slides ago, right? Kittiwakes, about 10% of those guys have plastics. We did the same study in fulmars where 90% of those guys had plastics. And I thought for sure that the fulmars were going to have higher levels of these additives in their livers as compared to the kittiwakes. But we don't see any difference. It's like, okay, so maybe plastic ingestion is not that important. The most important thing that we actually found here, though, that I didn't know what I was expecting, but it kind of came out as a bit of a surprise, is that we're finding levels in their eggs, which not only means so they're getting them, which is in their livers. You're like, okay, well, that's not all that surprising. But they're also transferring them to their eggs. And that's a bigger deal because a lot of these additives are hormone disruptors or endocrine disruptors. And you don't need a lot of endocrine disruptor at the embryonic stage to make a difference. And so these might look really low, but if you look at kind of past contaminants, you don't actually need a whole lot to be a bit of a problem. So we've done a lot more work on this, and this has been really triggered by the Stockholm Convention. So I'm going to try to summarize this, but in January 2020, the Stockholm Convention, persistent organics pollutant, a country, I believe it was Switzerland, nominated UV328 for consideration under the Stockholm Convention. As a government scientist, that kicked us up a notch to be able to provide evidence to either support or not refute, but inform that decision whether the government of Canada would support that or not, right? They wanted to have their own evidence and think about what does that mean and how do we tweak or not this type of work. And so again, with Xiaolu out of UCAR, we did this, and this actually just was published about 2 weeks ago. And so we actually were able to look at a range, and I believe there's 26 contaminants here. And these are all kind of plastic-related, either industrial antioxidants or UV absorbents and filters. And so the UV328 is this little set of bars over here. And this is the one that is specifically being considered by the Stockholm Convention. And so we've been digging into our archive and looking at this particular contaminant. You can see we actually detected it in all 3 of these species. There are some like UV326 that we didn't detect in any of our samples. Right? And then you have BHT, which has actually the biggest number of detections. Not only did we do all those contaminants, but we actually looked at these eggs from 1975 to 2019. So Environment Canada actually has a national specimen bank. And then in the long-term program that I inherited from my predecessor, and I'm currently the steward of, has been collecting eggs since 1975 from Prince Leopold Island in the high Arctic. And so every year we go to Prince Leopold Island, we get those eggs, we take an aliquot off, a little subsample to do whatever analysis that we're interested in that particular year. But the vast majority of that tissue then gets banked and archived at -40 at the National Wildlife Research Center. And so that allows me and others to say, okay, well, we didn't know to look for this one before. So we can go back into the bank, query all the tissues that we have, pull subsamples of all of those tissues, and then run a retrospective study just like this one. And so, again, we expected, for sure, I thought some of these plastic additives, we would see this increasing trend over time, right, that matches plastics. Wrong again, right? We see BHT is a little complicated, but we do see a declining trend over time with BHT. And then basically for everything else, we get these really spotty results, right? Some years we get detects, some years we get non-detects. And so this, and for those of you interested, we can talk details later. But this, it does show that UV328 is ending up in the Arctic in seabirds, and it's maternally transferred in eggs, and it happens in all 3 species. But it doesn't necessarily show us that these chemicals have any particular temporal trend. Why that is a discussion to be had still. But certainly this kind of adds to our understanding of what these are. And what this paper really did is really push us through, again, what the quality assurance and quality control needs to be for these plastic additives. Because how do you run plastic additives in labs full of plastics, right? There's a lot of method challenges in there. And so it really pushed us to really refine and think about this, especially when you have archived samples, right? Because what were they archived in? What was the treatment of that container? Okay, so UV328, just a little bit on the Stockholm Convention. All right, so it was nominated in 2021. I have my date off there for a second. Really interestingly, to be considered under the Stockholm Convention, you have to be persistent, you have to be toxic, and you have to have long-range transport. If you don't have long-range transport, you don't go into the Stockholm Convention. And the Stockholm Convention, you can have atmospheric transport, oceanic transport, or migratory animal. UV328 is actually the first time that anyone in the Stockholm Convention has invoked the migratory animal route as a long-range transport. And so this is something that has gone in and done a lot more work on. And so we've done a lot of work on wild species, and we've really kind of focused, I swear, the box is actually quite square, but these screens make it look, I feel like I'm getting seasick just like staring at the squiggly lines. But this has kind of really driven our work for the last couple of years, because Environment Canada negotiating staff, right, they sit at that Stockholm Convention table, and they need that information to either support statements or not support statements. And so this has driven a lot of our work of the last couple of years. Okay, moving on a little bit. There's also a good push, or a big push at the international scale. So we also work under the Arctic Council. The Arctic Council, up until March, when there was unlawful aggression against Ukraine, was the 8 Arctic nations. It's currently on pause, but up until then, there was an 8-country Arctic Council working group, and for the last several years, we have been working on different parts of plastic pollution. And 3 of the working groups, the CAF, which is the kind of biodiversity group, AMAP, which is the Arctic Monitoring Assessment Program, mostly climate change and contaminants, and then PAIM, which is the protection of the marine environment, all have plastics on their work plan. And so one of my roles, so I'm the co-chair for Canada for this group, and one of our works has been figuring out how these working groups work across each other in order to have monitoring programs, and again, what these policies and measurable targets are, what they should be, and then how do we figure out how to identify those, monitor those over time, so that when you have measurable targets and actions, you can actually see if it's working, right? You can make an action, but if you have no measurable target, who knows if it's working? Okay, so the last couple of years, what we've been doing is thinking about how do we monitor plastic pollution in the Arctic at the ecosystem level. So there's been lots of work at the UN level, at the G7 level, on monitoring in one compartment. We like a challenge at the Arctic Council, so we looked at 11 compartments. And so what we did, and this is the Arctic Council kind of definition of the Arctic. And so what we looked at is 11 different compartments, and basically made the first kind of ecosystem level assessment and plan. And so we have priority monitoring in four compartments: water, aquatic sediments, shorelines, and seabirds. And then these basically down here is where either they're low plastic or we just don't have the methods to do it, right? And this is a really big stumbling block. And this doesn't mean that they don't exist anywhere. It means that it doesn't exist at the pan-Arctic level of implementation. So if one country can do this really well, that's awesome, but we can't do it everywhere. So at the pan-Arctic level, it's not super, super useful. And so a lot of our work, again, prior to March, was working on this. These came out last year. And so if you're interested in this kind of big monitoring wide scale, there's the 4-page summary. This is the policy maker. This is like the glossy version you can like hand to a decision maker in the moment. There's also a 24-page monitoring plan. So this is more like what managers need to think about what they're doing or not doing. And if you're really into it, there's the 266-page technical guidelines, which at the working level, right, you can go and find out every detail of how to do it in the water, every detail how to do it in the sediments, every detail how to do it in the seabirds, right? And so when we're working at these really high levels that are really, really tied into these international policies, this is the type of spread of documents that are needed, right? There are these technical diagrams for the working level. There's kind of the synthesis, what I would often refer to as the management level. And then there's this really like, what does it all mean? Elevator pitch, 30 seconds in a glossy document, right? And which is what goes to the decision makers and policymakers. So, okay, I'm going to tie this into climate change because this is also a big thing that we're working on. So one thing that you may have noticed is that ice and snow is way down here, right? This is because it doesn't really have a great method, right? We don't have great methods that are widely applicable, widely implementable across the Pan-Arctic. But that doesn't mean it's not important. In fact, there's been a number of studies now that have kind of thought about these atmospheric inputs, right, into all kinds of different places. And one of them is snow. And so this is actually a recent paper in Nature Reviews that Melanie Bergman led with a group of us, where it's been estimating, how much microplastics is in snow, how much is in sea ice. And you can see that, potentially, depending on your unit, these are some really big numbers. But it's very, very difficult to, again, do this at a level of monitoring, right? Not just research, I got a snow sample, look at these amazing levels, but how do you do it repeatedly over time across huge landscapes? But this is just an example. This is actually a recent paper out of McMurdo in the Antarctic, and you can see all kinds of the plastics that they were finding in the snow. Pathways are, work in progress, but this is happening. So if we tie it back into climate change, right, there are some big consequences for plastic pollution and climate change. One of them, right, climate change, warming temperatures, melting glaciers. I was actually just watching, what was it? It was this morning, like the Today Show. It was some like morning show. And you know what they had? Melting glaciers. There's this huge thing where right now where a whole bunch of the glaciers in the Rockies are actually disappearing, right? This is not news to probably anyone in the science community, but I was actually impressed. It was on the Today morning show. And so this is well documented over time. I mean, people have spent their lifetimes documenting these changes in some of these ice sheets, right? But one of the challenges is what does the melting glaciers and the melting sea ice mean for plastic pollution released into the environment? And so if you have atmospheric deposition and it's then locked in those glaciers, and has been for years and years and years, and now those glaciers are melting out, where are all those microplastics going? So this is a big question that's outstanding and that we're really interested in. And this is both in freshwater environments as well as marine environments. All right, this is gonna seem like a bit of a left turn, but this is one of my favorite old school projects. Who heard about the rubber duckies in ever, actually? A few people, right? So this is one of the most amazing natural experiments, and Charles Ebsmeyer is famous for this. So in 1992, this is going to seem left field, but I swear I'll come back. So in 1992, there was a huge tanker, and it was en route from Hong Kong to Tacoma, and one of those big shipping containers fell off the side, as it happens all the time, but this shipping container was full of rubber duckies. And it wasn't just any rubber ducky. It was a specific brand of rubber ducky that didn't have a hole in the bottom. So they weren't squeaky, they were just rubber, right? But that means they didn't take on water and then sink. And so what Charles Ebsmeyer did is he figured out that he could actually understand a lot about ocean currents by figuring and tracking out where these rubber duckies, this very specific type of rubber ducky, actually landed. And so he put out a whole bunch of media calls and all kinds of things. You have to remember it was the 90s, right? There was no internet, there was no blogs, there was no Facebook. But he was able to track rubber duckies. They started landing on the coast of Washington in 96. They showed up in Alaska in 92. All kinds of places they showed up. What's fascinating to me is they actually started showing up in the Atlantic about 10 years after they were just, fell into the ocean. And so these, even in the early days, when, and this is plastic, right? This was seen as kind of an inert thing moving through the ecosystem that we could track. And so we've always known that the Pacific and Atlantic were connected, specifically from a debris perspective. But this kind of questions, how does it move through the Arctic? Because if you go back to the 90s, right, this is this time period, this is 79, we don't have really a 90s one, a lot of the Arctic is still frozen, right? And so one of the things that people are speculating right now is as we kind of move from this kind of thicker, more dense sea ice through, again, probably no surprise, right? We see lower and lower and lower levels of sea ice throughout the year. Now to the fact that we actually have open water in much of the kind of Beaufort area that we didn't really ever see before. What does this actually mean for plastic pollution that floats and can travel across the Arctic? And so this is one of the reasons why we put shoreline litter on the monitoring program, because there is an expectation, specifically with the North Pacific Garbage Patch and the Atlantic Garbage Patch, that we actually might see, because right now in the summertime, these are actually in winter, you can actually go all the way across, right? There's some sea ice, but you actually can make it across, right? Cruise ships currently going across the Northwest Passage in the summertime. And so there is a thought that now that that summer sea ice is reduced, you potentially are going to have litter moving up and into the Arctic where, because it was positively buoyant, right? It's at the surface, that sea ice was preventing it. And as that summer sea ice retracts and retracts and retracts, that litter basically can just float on through. And so this is actually a really big concern moving forward. The other thing that we were talking about earlier, and I have kind of inadvertently stumbled into, is the world of bottom paint. And so in 2018, we did a bunch of sampling in a national wildlife area. Again, Environment Canada, government scientists. One of the things that I'm tasked with is working with partners to understand the ecological integrity of protected areas. And in this particular case, it was a national wildlife area. And so we did a bunch of sampling, and two of my grad students one day called me, and you know it's never good when two of your grad students are calling you into a meeting one day. And they showed me this, and they were like, what is this? What do you think this is? We can't figure it out. And so we've kind of gone down this path, and you can see they're, kind of uniform in color. We kind of batted it back and forth. We ended up on the phone. We have both ROM and an FTIR, polymer identification. And basically what we came to through talking through a lot of people is that this is actually bottom paint. It's the anti-fouling, self-ablating bottom paint that they paint on the bottom of vessels, right, to prevent invasive species from moving around on bottoms, right? And it's called self-ablating because over time, and I can say this as a boat owner, you put it on, right, and it sloughs off. So as things attach to it, it either chemically prevents things from attaching to it, or if things attach to it, it kind of sloughs off, right? It's designed to slough off into the environment. Enter into the environment, enter microplastics. And so this actually is a big thing that's emerging in the literature right now. There's all kinds of additives in there, both plastic and metals. We have a new project actually starting this year. We have 2 communities in Nunavut that we're working with. They're doing water sampling. It's, again, a part of an ecological integrity work that we're doing. In Transport Canada is the government or group that's kind of been tasked within the government of Canada, just kind of thinking about this at the regulatory level, right? So bottom paint is useful because it's being used to protect against aquatic invasive species. So how do we still do that and not create microplastics in the environment? And so this is actually work we have funding starting this year for the next 3 years, and we're going to be doing a lot more of this work. And so this is a really kind of exciting, I think, time to be thinking about this. And again, this is a policy window that I think that we might be able to actually do some really good stuff on. Okay, and then, why does this matter in plastic pollution, and why does this matter in climate change? Well, again, not my particular field, but I have a lot of colleagues who think a lot more about this. And climate change, again, this reduction of summer sea ice is actually leading to increased shipping. And so why we're working with those 2 communities in Nunavut is specifically they're anticipating a huge increase of shipping going past their communities because of the opening of the Arctic archipelago as a quicker route between continents, right? And so there's, this is just a couple of snapshots of things that people have done. This is 2013. You can get, there's something called AIS, the Automatic Information Systems. We actually have the number of ships going through time. We can track that. Jackie Dawson at the Ottawa U is kind of the guru of that. So we can look at all the automated information systems. We can actually track vessel traffic over time. You can see a change in 2013 and 2019. Same thing. And so there's some projections here that we're going to see more routes, and they're going to be more confined. They're going to have these very strict routes. And so some communities, will have no change. So example, Hudson's Bay, right? You're not going into Hudson's Bay unless you're going into Hudson's Bay, right? But there are other communities, especially along this line of Baffin, which is actually right where I work, that are going to experience a ton of projected increased traffic. And so this is a big concern currently that they're really we're trying to grapple with. And this idea of, understanding now and later, we don't have a time machine. We can't go back and get those samples. And so a lot of the work that we do in Nunavut is trying to get a hold of what's happening there now so that we can understand those impacts. And again, this is all linking into this plan. So again, if you're kind of interested in these types of things, I really encourage you to look at these documents. Okay, so the next step for plastic pollution stuff in Canada. How many of you look at the Minister of Environment mandate letter? What, just me? No one else? Oh, thank you. One other person. If you're a federal employee, you always look at the mandate letter because it's your marching orders for the next, depends on the election cycle, years, right? And so I encourage you to go and look at the mandate letters. In our mandate letter, there is 8 direct points on plastic pollution. 8. That's crazy. Sometimes there's like 1 for protected areas and there's 1 for cooperation. We have 8 specific points. And so this is huge. So for the last 12 months, one of the things that we've been working on is the renewal of the plastics funding. So we had funding, I guess it was 4 years ago. It just ended in March 2022. And so for over a year now, we've been working on the renewal of this plastic funding under the federal budget. And so actually 2 weeks ago, we got the Treasury Board all like buttoned up and okay, and I got my code funding and all this stuff. So we now have renewed this now for the next 5 years. And so this is a pretty exciting time. So currently, Environment Canada researchers and strategic policy branch and all the fun different groups are trying to figure out kind of what did we do in the last one? What do we still need to do? What questions have we answered? What do we still know nothing about? And so this is all the work that we're kind of thinking about. And so currently there's a national strategy for monitoring plastics pollution in the environment underway. And we're going to be doing a lot of the synthesis work and understanding and what have we learned and how does that change our plastics agenda moving forward, right? What are the things we can take off and what are the things that we were like, didn't get to that at all. And so that's kind of what is driving my program right now as we think moving forward about, again, where do we need to be going on this and what the policy windows are, right? So it's figuring out what those times are. And I think with that, I'm going to stop. You have been an excellent audience. I thank you greatly, and I'll take any questions if you have them.
Thanks so much. Sorry, you can't see my big smile under there.
That's okay.
That was awesome.
Your eyes were smiling.
Thank you so much, Jen. That was a fantastic lecture, and we do have time for some questions from the audience.
They want you to come.
I will
There's questions. There's time for the
Time
questions.
for questions. Yes, Chi-Ching.
Hi, Dr. Provencher. Thanks so much for your excellent presentation. I really like it. I have one question about the additives you have mentioned about the antioxidants and UV absorbents. It's very surprising they're declining, right? Is it possible that there are new emerging antioxidants are used or some other reasons?
Yeah, I'm going to repeat the question so that they can hear. So and you can correct me if I get this wrong. So it was surprising that there was a decline in those antioxidants, and the question is whether they have basically replacement or new antioxidants that are being used that we didn't detect. Is that the question? Yeah, it's a great question, and the answer is I have no idea. Why there was none. So BHT is a little complicated, and I encourage you to read the paper because Jalou, who is kind of the environmental chemist that I lean heavily on for some of these things, BHT can be metabolized, and so there's some question about why there are some patterns there. Whether or not they're using replacement or alternative, this is the bane of the environmental chemist, right? Derek Muir, who is an amazing research scientist, environmental chemist, talks about this, right? They are, I can't remember the exact stat, but it's 30,000 chemicals added to the books every year. As an environmental chemist, you are how, why, how do you do that? And so we actually have a project with Roxana Schuring. She's a chemical engineer at, not Ryerson, Metropolitan Toronto University is the new name, who is going to do some broad screening. But that is, and so one of the things that I'm always really willing to share is and several colleagues in this know this, I can get tissues really easily, but I don't have every machine to test for everything, right? And so I have a ton of colleagues that I send tissues to because it takes an army to think about all of the things you could possibly screen for. And so the answer is that it's we often have to go in with some kind of a priori, what are we going to screen for? And so in this case, we chose those because UV328 was listed by the Stockholm, and Jalou can measure them, right? But then there's a whole other suite of organophosphate esters that Roxana does. And so, some people look and they're you publish a lot of papers. It's I actually just share a lot of tissues because I know I can get the tissue, but there are so many other people who can take those tissues in so many different directions that I can't possibly do. And so that's where the collaboration really comes in. And so if there's other chemicals to test for that are emerging, always, but how to test for them is always the challenge. Any other questions?
Miguel, and then come back.
Yeah, going back to your polymer study, I'm just curious, what's the variability of population sizes between those different, communities, and what are the implications of that for your results, and how does it play into why you think those areas are more polluted, or the organisms have more plastics?
Okay, so I'm going to read the question, then you can test whether I got it right. So your question is, how do the fulmar population size vary across these sites, and how does that influence how much plastics they have?
Yeah.
Okay, so the tricky part is that for those of you who don't spend all your time with your nose in a bird book, the orange is where they breed. The purple is where they found all year, but out at sea, and then the blue is the non-breeding. And so the other tricky piece, right, is that Prince Leopold Island is a breeding colony. And so I know where those birds are. On Labrador Sea, there's, technically it looks like they breed here, but the birds that we catch are out at sea. And then we also have birds out at sea here, but they're actually from fisheries. And through genetics, we know that they actually are European nesters. And then on Sable Island, it's in the winter time, and same in Vancouver, they wash ashore. And so where they breed is a mixed bag. And so we know that the birds on Sable are a mix between Arctic Canada breeders and European breeders. Because this map is deceptive, right? It's not like they only breed in the economic exclusion zone of Canada, right? The range is actually circumpolar. And so we don't think, so the population sizes, I could give a whole nother talk on fulmars because they're fascinating, and we've done a lot of current work on them, but we don't, okay, and then there's like these orange dots, which are fascinating, but we actually think that this is a weird sink population, and they come over from Europe. So these dots are not related to any of these guys. So how population size, I don't think population size affects how much plastic pollution they have, but I do think population size affects how well we can monitor them, right? And so if in these locations we only get beach birds, so do the beach birds have a winter signal that is higher because of where they're wintering? And do these birds have a lower signal because they're in cleaner waters, because they're foraging locally? I think the answer is yes, but that's the whole point. They're environmental indicators, right? And so as the birds move through and move down, and we've done this where if you get the birds early in the year, if you're kind of like sitting on the cliff and they arrive, I'm being a little facetious, but like if you get them early and then you wait 2 months and you catch the same bird again, we've done this where you catch like murres as they arrive versus murres as they like have hung out there for a little bit, it's a seasonal decline. And so they're somehow getting rid of their plastics. And I think it's because they're grinding up and pooping it out. And so I actually do think where they're breeding and where they're eating is reflecting where they are. But in this case, that's what we want, right? Because it's actually about tracking plastic pollution in the environment.
We have time for maybe one quick question.
Sure. This might be a super naive one because I don't know anything about ocean currents or bird migration.
What?
Are there similar processes going on in the Antarctic? Like, do we have a second polar replicate to look at some of these patterns?
Yeah, so great question. The problem is that the Antarctic and the Arctic have completely opposite atmospheric and oceanic properties. So the Arctic Ocean is water, Antarctic is land, right? The Arctic is water surrounded by land with only a few inputs, whereas the Antarctic is this island with this like swirling current around it. And so there is, I belong to it's SCAR is the Scientific Committee for Antarctic Research, and we have a dual polar program, and it's like completely different. And so what happens in the Antarctic, so in the Arctic, you have this throughput. So typically what happens, like the rubber duckies, right? They come up through the Pacific is like slightly higher, and it comes down into the Atlantic. In the Antarctic, there's a circum-Antarctic current, and there's an upwelling zone. And so interestingly, if you're sampling north of that, you find plastics. And if you sample south of that, there's limited plastics. And so there's like this circum-Antarctic upwelling zone that appears to be keeping most oceanic current out. But atmospheric deposition is still like real. And there's a few programs that I work with. There's one in National Geographic where they're actually tracking plastic pollution from tourist boats. So you actually have gray water discharge coming out of those boats. So you have these boats that are not only sloughing off their bottom paint, but you have gray water sewage being offloaded as well. And so potentially that's the bigger route to the Antarctic.
Well, thank you so much.
No problem.
Can you please join me in thanking the speaker?
Yeah, no problem.